ongoing series on plant hormones, we have already discussed about auxins and gibberellins. In those lectures, I gave you an overview about the historical development of auxins and gibberellins, some of their important properties, how they are biosynthesized in the plant cells, what is their signaling pathways, how they exhibit the effects that are being produced by these hormones, and then in the last we discussed what are the various effects, physiological as well as biochemical effects that are produced by auxins and gibberellins. So friends, in continuation, today I am going to start with another important class of plant hormone, that is cytokinins. Now cytokinins are the hormones that are responsible for the growth of the plant. Specifically, they cause induction of the cell division. They increase the cell division, thereby increase the cell size and increase the size of the plant. And that is how they induce the growth in the plant. Now, it all began in 1892 when J. Weissner, who gave the hypothesis that there are certain endogenous factors that induce the cell division in the plant. But he also stated that proper balance has to be there between those endogenous factors in order to initiate the cell division in the plant cells. Later in 1913, Haberland, he identified or he isolated certain diffusates from the phloem tissue. He was not able to elucidate the exact compound, but he identified a crude form of extract or diffusate from the phloem tissues that were responsible for inducing the cell division in the parenchyma tissue of the potato tubers. So this was another instance where the scientist came into light about certain cells that are also present in the vascular tissue that will induce the cell division. Following his work in 1941, Johannes Wien Overbeek, he also worked upon a milky endosperm from the coconut and found that this milky endosperm or the coconut water is also responsible or has the ability to stimulate the cell division. So such kind of experiments were going on throughout the globe in order to identify these components, because Weissner was the person who approximately 130 years ago devised or hypothesized that there are certain factors that are responsible for cell division, and after his hypothesis, scientific community start working in this direction. And a lot of work was going on, and these were the few key discoveries that were made in the process of discovering these important hormonal compound, that is cytokinin. Then in 1954, Jablonski and Skoog, they followed the Haberland experiment and showed that whenever a piece of vascular tissue that was cultured on the top of tobacco pith tissue induces the cell division in pith cells, and normally we know that the pith cells, they do not have capability to divide under the normal circumstances. So in other words, we can say that the experiment of Jablonski and Skoog, they supported the hypothesis of Haberland where he proposes that the phloem tissues, the diffusates from the phloem tissue, have capability of inducing the cell division. So this is basically extension of the Haberland's experiment. But it was Miller, Skoog, Salza and Strong in 1955 were able to first identify the first cytokinin, and they named this compound as kinetin, or chemically it was 6-furfuryl amino purine. The name kinetin was given because this compound was found to induce cytokinesis, that is cell division. And they isolated this compound from an autoclaved sample of herring sperm DNA. Later, Skoog also tried to repeat this experiment, but this time he took fresh sample of herring sperm DNA, and to his surprise, he was not able to isolate the same compound kinetin. He repeated the experiment again he was not able to find kinetin from the fresh herring sperm DNA. So he again repeated with the autoclaved sample then, but this time he was able to isolate kinetin. So he again gave the hypothesis that kinetin, the first cytokinin which they isolated, is nothing but an artifact that was produced due to certain chemical modification when the sperm DNA sample was autoclaved. Or perhaps this was heating of the sample that leads to the chemical modification and the production of kinetin in the sample DNA of the herring sperm. Now later in 1961, C.O. Miller from Indiana University, he was the person who first isolated a cytokinin compound from the plants. He was working on the maize plant and he isolated the compound that was responsible for the cell division. But he was not able to identify or elucidate the structure of this particular compound. So following his lead, in 1963, D.S. Latham, they again start working on the maize plant, and they also isolated a compound which they also identified and named that as zeatin, because the compound was isolated from maize, that is zea maize, so they named the compound zeatin. So Latham was the person who first elucidated the structure of the zeatin, but Miller was the person who first isolated the compound but was not able to identify that compound. Since both these persons were having a significant contribution in the discovery of the cytokinin first cytokinin from the plant, therefore Miller and Latham are credited with the discovery of zeatin, which is the first cytokinin from the plant cells. Now zeatin occur in both cis as well as trans form in nature, and trans zeatin has been found to be biologically active, whereas cis form was found to be inactive or was having very less activity in the plant cells. And these are the few examples of certain cytokinins that have been isolated from the plants. Until now, more than 200 cytokinin compounds have been isolated from different plants in the nature. And as you can see the structure of these naturally occurring cytokinins, you will see one thing common in all these structures, that is the presence of the adenine nucleus. And therefore the cytokinins are the compounds, these are the compounds which are defined as naturally occurring adenines which possess either an isoperinoid or aromatic side chain at N6 position, and these compounds are capable of promoting cell division and exert other growth regulatory functions. So as I told you initially that cytokinins are the compounds that are responsible for the growth of the plant. They are responsible for the cell division that leads to the growth of the plant. Now apart from these adenine derivatives which are naturally occurring cytokinins in the plants, there is another class of cytokinins that has been identified in nature, and these are the phenylurea type of cytokinins. And one of the most common example of phenylurea type of cytokinin is the diphenylurea. But these types of cytokinins have not been reported from the plant, though they occur in nature, but these have not been reported from the plants. Now cytokinins, they exist in plants not only in the form of free bases, that is as such zeatin, but these are also found in the form of glycosides whenever sugar molecule is attached, specifically in the form of nucleosides when a riboside is attached to them, or they are also present in the form of nucleotides when the ribose sugar is further having phosphate groups attached with them. So we can say that in plants cytokinins occur as such in the free base form, or they occur in the form of glucosides, specifically nucleosides when ribose sugars are attached, or in the form of nucleotides when the phosphate groups are present in those ribose sugar molecules. Now we move on to the biosynthesis of cytokinins, that is how they are produced, how they are synthesized within the plant cells. Broadly, there are two different pathways that have been suggested for the biosynthesis of cytokinins, and first one is the isopentenyl adenine dependent pathway, and the second one is isopentenyl adenine independent pathway. Now since cytokinins are responsible for the growth of the plant through cell division, these cytokinins are found in larger concentrations in certain developing tissues where the cells are vigorously dividing, that leads to the growth of the plant. And such organs or the tissues are the root tip. We know that root tip is the area where the cells keep on dividing. Similarly, shoot apex is there. The cells keep on dividing at the shoot apex, thereby increasing the overall length of the plant. Cambium cells keep on dividing and increases the girth or the size of the plant. And likewise, immature organs are there which keep on developing with the growth of the plant, and the cells keep on dividing there also. So these are the areas where the concentration of cytokinins was found larger in comparison to the other parts of the plant or the other tissues of the plant. Now we move on to the isopentenyl adenine dependent pathway. Now initially it was found that the isopentenyl tri or diphosphate are produced from a common precursor, that is dimethyl allyl pyrophosphate or DMAPP, which is produced through a mevalonic acid pathway. Now this DMAP is acted upon by an enzyme, adenylate isopentenyl transferase, thereby producing isopentenyl adenine di or triphosphate. If ATP is involved, then we get triphosphate. If ADP is involved, we get diphosphate. And this DMAPP can directly get converted into isopentenyl adenine monophosphate if adenine monophosphate molecule is attached. Later this undergo hydroxylated of cytochrome by cytochrome P450 enzymes, thereby producing trans-zeatin riboside monophosphate. So isopentenyl tri and diphosphate, they are acted upon by cytochrome P450 monooxygenase enzyme, thereby converting the isopentenyl triphosphate or diphosphate into trans-zeatin riboside monophosphate. And this ribomonophosphate trans-zeatin is acted upon by the log enzyme, which are also known as lonely guy enzyme, and they get converted into trans-zeatin. Now this interesting name lonely guy was given to these enzymes because these were first isolated from the rice mutants, which contain only single stamen and no pistil. And since only single stamen is present, the name lonely guy was given to these enzymes. Now in isopentenyl adenine independent pathway, the trans-zeatin type of cytokinin is largely produced directly by the transferring of hydroxylated side chain from the precursor, that is hydroxymethyl butenyl diphosphate, to the adenine ring. So this completely excludes the formation of isopentenyl adenine derivatives, and therefore it is largely known as isopentenyl adenine independent pathway. So here you can clearly see the two pathways. On the left is the isopentenyl adenine dependent pathway, wherein mevalonic acid pathway produces DMAPP, that is dimethyl allyl pyrophosphate, which is acted upon by adenyl isopentenyl transferase enzyme using either ATP or ADP molecule, thereby giving rise to isopentenyl adenine di or the triphosphate. And this di or triphosphate derivative is further converted into monophosphate derivative. This monophosphate derivative can be directly produced from DMAPP by the action of, again, the similar adenyl isopentenyl transferase, because isopentenyl transferase enzymes basically they transfer the isopentenyl moiety of DMAPP onto the adenine ring, because cytokinins are nothing but these are the adenine derivatives, and this monophosphate then is acted upon CYP450 enzyme and gets converted into trans-zeatin riboside monophosphate. Now you can also see that these isopentenyl tri or diphosphate can also be acted upon by CYP450 enzyme and can get converted into trans-zeatin riboside tri or diphosphate, which eventually again gets converted into trans-zeatin riboside monophosphate. And this monophosphate is acted upon by the log enzyme or lonely guy enzyme and get converted into trans-zeatin. And this trans-zeatin, once it's produced, when it is released in the plant cells, it undergo metabolism or it undergo glucosylation, wherein a glucose molecule gets attached to the cytokinin molecule, and this glucosylated form act as the storage form of the cytokinin. Whereas in case of isopentenyl adenine independent pathway, you can see the precursor, that is hydroxymethylene butylene diphosphate, it transfers its chain onto the adenyl ring with the help of adenyl isopentenyl transferase enzyme, and thereby producing trans-zeatin riboside triphosphate, di or triphosphate, and similarly it can also produce trans-zeatin ribo monophosphate, which again is acted upon by log enzyme and thereby producing the trans-zeatin molecule. So this is basically how the trans-zeatin, which is the most important or the cytokinin that is found in the plants, or the major cytokinin found in the plants, is biosynthesized in the plant cells. Now we move on to the cytokinin signaling pathway and how this cytokinin produce their effect on the physiological or the biochemical aspects of the plant. Now this signaling pathway or the signaling system is normally considered as the two-component signaling system, which is also known as histidyl to aspartyl system or histidyl aspartyl phosphorylase system. Now the term phosphorylase is given because during this signaling pathway, the phosphate group is passed on from one component to the other component to the other component, finally eventually leading to the activation of the cytokinin transcriptional factor, which increases the expression of the genes that produces the effect related to the cytokinins. And since the phosphate group is passed from one factor to the other factor in a form of a relay, therefore it is known as a phosphorelay system. Now, this kind of system has been found in bacteria, in fungi, as well as in plants, but has not been reported from the animals. Now, the first component of the signaling system are the cytokinin receptors, which are also known as CRE1 or cytokinin histidine kinase receptors, and when they are present in Arabidopsis, they are known as AHK2, 3 and 4 receptors. Now, these receptors have a specific configuration or specific domain, which is known as cyclase histidine kinase associated sensing domain, or the chase domain, which is present on the outer side of endoplasmic reticulum. And this chase domain is important because it is the place where the cytokinin binds to its receptors. Since chase domain is the part of these receptors, it is responsible for the binding of the cytokinin on the receptor, which initiates that phosphorelay system, that is the transferring of the phosphate group from one component to the other component to the last component, thereby activating the transcriptional factors and eventually producing the cytokinin induced effects. The second component are the response regulators, and as their name indicates, response regulators, these are the components that induce the response in the nucleus of the plant cell. So, as soon as the receptors get activated by cytokinin binding, the phosphate group transfers through the phosphorelay system, eventually activating these response regulators. And depending upon the properties, these response regulators are of three types: type A, B and C. Now, type B is the major response regulator of cytokinin because it is having a transcriptional factor domain, which is known as GRAP. Whereas type A and C are devoid of this GRAP transcription factor domain. Now, whenever the type B response regulator get activated, it leads to the activation of various transcriptional genes that leads to the cytokinin induced effects, and therefore type B is normally considered as a positive regulator of the cytokinin response. This type B response regulator also activates type A response regulator, which produces other effects, though it does not have a transcriptional factor domain, that is GRAP, but it also it induces other effects in the plants. And vis-a-vis, it also acts as a negative feedback regulator for the response regulators, because as soon as sufficient amount of type A get activated by type B, it sends its signals back to the response to the phosphorelay system, thereby preventing the transferring of the phosphate group and thus preventing or inhibiting the cytokinin effects. And that is why type A is always considered as a negative feedback or the negative regulator of the cytokinin response. Now, how it happens? So, whenever the cytokinin binds to the chase domain of the receptor, they bring certain conformational changes, such as causing the dimerization of the receptors. And once these receptors get dimerized, they undergo autophosphorylation. That is, the phosphate groups start attaching to the histidine group of these receptors. Now, this activates the phosphorelay system. And in this, the phosphate group from the cytokinin receptors, they transfer to the aspartic acid residue of the receiver domain, because histidine takes the phosphate group when the receptor undergo phosphorylation, and from this histidine group, the phosphate group then moves on to the aspartic acid residue of those receptors, which act as the next receiver domain. So this is basically the first step of the phosphorelay system. Now, the phosphate group from the receiver domain, that is the aspartic acid domain, it gets transferred to the intermediates, and these intermediates are nothing but these are the histidine phosphotransfer proteins, or HPTs or AHPs 1 to 6, and in these 1 to 6, AHP6 is a pseudo phosphotransfer protein because it does not have a histidine group to bind the phosphate group. Now, once the these histidine phosphotransfer proteins, they take up the phosphate group, they pass on this phosphate group to the next component of the phosphorylase, that is response regulators, which are present in the nucleus. And from there, various response regulators, specifically type B, gets activated, which causes the transcriptional factor activation and activation of the genes, vis-a-vis also activation of type A. So in this diagram, you can clearly see that how cytokinin binds to the chase domain, where thereby activation of the receptors, they undergo phosphorylation. The phosphate group from the histidine transfers to the receiver domain, that is aspartic acid domain, which is indicated by D. From this receiver domain, the phosphate group is transferred to the histidine proteins or AHPs. From AHPs, these are transferred to the response regulators, which are present in the nucleus. These response regulators activate type B specifically, which is having a transcriptional factor GRAP, and this transcriptional factor further activate various genes. Various genes or these genes are upregulated to exhibit the effects that are induced by cytokinin, and they also activate or upregulate type A response regulator, which is a feedback regulation, which act as a feedback regulation for the cytokinin actions. Now we move on to the various actions that are exhibited by cytokinins. So as I told you that cytokinin is a growth hormone and it induces the cell division. So cytokinins stimulate the cell division during embryogenesis in the shoot apical meristem, young leaves, the cambium and the cultured plant. So when they induce the cell division in the shoot apical meristem, they causes increase in the length of the plant. When the cambium cells, the cell division is activated by cytokinins, they basically increase the girth of the plant. When the young leaves cell division occurs, the size of the leaves also increases, and they, in various tissue culture studies, they have shown to increase the cell division in the plant cells, in the cultured plant cells, thereby producing the callus or producing the calluses or producing the group of in the suspension cultures. Then cytokinins also stimulate the growth of lateral buds and they lead to the release of the apical dominance. Now we know that auxins are responsible for the production of the apical dominance, because auxin is a hormone which leads to the apical dominance, thereby suppressing the lateral growth of the plant. Whereas cytokinins, they stimulate the lateral growth and release or inhibit the apical dominance, and therefore we can say that cytokinin antagonize the actions of auxins, and as I discussed, that auxins produced in the shoot inhibit the release of apical dominance, that is, they carry on the apical dominance in the lateral buds, so they do not allow the lateral growth of the plant, whereas cytokinins, they induce the lateral growth, thereby inducing the increase in the girth of the plant, increase in the width of the plant, and they release the apical dominance, or in simple words, they inhibit the apical dominance. Now it has also been observed whenever the apical bud is removed from the plant, the release of cytokinins in the lateral buds increase, which causes the formation of new vascular connections. And when new vascular connections are formed, they initiate the growth of the lateral bud, thereby releasing the or inhibiting the apical dominance or the apical growth. And this is how basically the lateral bud growth is stimulated by the cytokinins. Now as I told you that cytokinins also cause increase in the cell division in the leaf tissue, thereby causing the cell expansion, which also results from the enlargement of the cell. So as cell divide, they also enlarge, and that leads to the expansion of the leaf. Now cytokinins also control the flower and fruit development, and they also regulate leaf senescence. How they do it? The cytokinins basically increase the mobilization of the metabolites, and they stimulate the synthesis of various proteins and nucleic acids in the leaves, vis-a-vis also in the flowers and the fruits. And when the accumulation of metabolites increases, or the mobilization of the metabolite increases, and the synthesis of various proteins increases in fruit and flower, their development occurs. When the synthesis of nucleic acid and protein increases in the leaf, their senescence decreases. So they prevent the leaf senescence also. Now cytokinins has been found that whenever they are applied on the yellow leaves, they also cause regaining of the chlorophyll. That is, they convert those yellow leaf back to the green. and they thus preventing the leaf senescence. They also stimulate the vascular morphogenesis during embryonic development, that they help in differentiation of the vascular tissues into their components. Cytokinins also play an important role in plant pathogen interactions, and the most common example of this interaction is the Agrobacterium tumefaciens, which is nothing but a soil bacteria. And this bacteria is responsible for causing the tumor gall disease in the plant. So whenever the plant is injured, this bacteria infects the plant and it leads to the production of tumor gall disease. Now, if we look into the DNA of this Agrobacterium tumefaciens, you will find that it contains a plasmid which is known as TI plasmid, that is tumor inducing plasmid. And this plasmid has a tumor inducing DNA domain. Now, this TI DNA contains certain genes which are responsible for increasing the production of cytokinin in the infected plant. And once the cytokinin production is increased in the infected plant, there is uncontrolled cell division that leads to the tumor gall disease of the plant. So this is just one example of interaction of cytokinin or the plant or the pathogen interaction, how the cytokinin play a role in a plant pathogen interaction. Now, cytokinins also stimulate the morphogenesis in case of shoots, and it has been observed in the tissue culture studies, wherein they initiate the formation of bud, and once the bud is initiated, they also lead to the growth of the bud by virtue of their effect on the cell division. Now, whenever the cytokinins are used in higher concentration, it has been found that they generally inhibit or delay the root formation, and they inhibit the root growth and promotive effect of the auxin on the root initiation, because this is another example where cytokinins antagonize the actions of auxin, because we know that auxins causes the induction of the root growth or the root formation, whereas when cytokinins they are applied in higher concentration, they delay the root formation, they inhibit the root growth. Now, whenever we use auxins and cytokinins in combination, they have shown certain physiological actions, and since cytokinin has an antagonizing action to auxin, so the resultant effects will depend upon the ratio in which we are using both cytokinin and auxin. If cytokinins are higher, then they will have an antagonizing effect to auxin. If auxins are higher, then they will have an antagonizing action of the cytokinins. So friends, this was about cytokinins, how they produce their effect, what is their signaling pathway, and what are the various effects that are produced by cytokinins in the plants. Thank you.